Good evening, everyone. I'm glad to see our on-time and late arrivals. It's nice to have a good crowd in the auditorium for this really important tradition here at the MBL. My name's Sebastian Lay. I work here at the MBL, and I just have a few housekeeping items. Many of you know these by heart. First, please, 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 either turn off, silence, mute, airplane mode your cell phones. Otherwise, we're going to throw them in the old pond. Or feed them to an octopus. No, we won't do that because we care too much about our cephalopods. Second, this lecture will be recorded and is being simulcast on Zoom, and so there will be some questions from online as well as from the auditorium at the end of our talk. If you don't have an opportunity at the end of the talk, there's usually about 10 minutes or so left. There is a reception following on the back terrace of Swope, where you'll get to interact with our speaker this evening. And then finally, it's my pleasure to introduce the introducer this evening, Tatiana Piotrowski. She's an investigator at the Stowers Institute for Medical Research, which is a wonderful institutional partner for the MBL. And importantly, she's here to teach co-lead the Zebrafish module in the embryology course right now. And so a very appropriate introducer this evening. Tatiana, thank you.
Thank you, Sebastian. And Len, I have to apologize. I had to really shorten your CV because otherwise we wouldn't be able to hear your research. So it's my great pleasure to introduce Len Zahn. He's a Grousbeck Professor of Pediatrics at Harvard Medical School. He's also Professor of Stem Cell in Regenerative Biology, and he's been for many years an investigator with the Howard Hughes Medical Institute. So the science community had also the great joy of recently celebrating his recent election to the National Academy of Sciences, and I think we should give him a hand for that. And so Len is a clinician scientist, and he was trained in hematology. And in addition to performing stem cell transplants on patients, he also developed the zebrafish into a model to understand blood disorders and blood development in zebrafish. So he pioneered the use of zebrafish as a disease model by identifying the genes that cause blood disorders in zebrafish mutants. And by doing so, he actually identified 5 novel human diseases. So in addition to his work on blood development, he also developed the zebrafish into a model of cancer, specifically melanoma or skin cancer. And 4 of his discoveries already moved into clinical or phase 1 clinical trials. There's one small molecule that he identified, which is called prostaglandin E2, which regulates stem cell biology or stem cell engraftment, and this has been already now used in the clinic for hematopoietic stem cell engraftment. And this molecule has been already given to 150 people. So this means really that Len's research has life-saving impacts. So in addition of being a brilliant scientist, Len is also a terrific advocate for the scientific community in large. So he's the founder of the International Society for Stem Cell Research and also for the Zebrafish Disease Model Society. He was heavily involved in convincing the NIH to get the zebrafish genome sequenced. By doing so, he became the director of the Trans-NIH Zebrafish Genome Initiative. And his lab also helped us really develop and generate maps, which allowed us to map all these zebrafish mutants. So as you'll hear from his presentation, he has an unbound enthusiasm and infectious enthusiasm, I have to say, for science. And if you go to conferences with him, you'll often hear his laughter in the poster sessions, where he really still attends all the posters, which is fantastic, I think. So all these attributes make him a fantastic mentor. He has won many mentoring awards. So far, his lab has trained 37 grad students and more than 60 postdocs. And many of them have gone on to establish their own labs by now. So as you can tell from my introduction, Len has achieved really a lot of accomplishments in his life, including authoring 500 papers. But recently, his latest achievement that he's very proud of is actually outside of the field of science. So he's an accomplished and gifted trumpet player, and he told us that COVID gave him ample time to practice trumpet. And he's very proud to announce that he's now able to play the double high C note. So So Len, on behalf of the MBL, the embryology course, and also everybody here in the audience, I would like you to invite you to come up to the podium, and we are very much looking forward to learning about your latest research.
Thank you. Super good. Well, thanks very much. It's really a treat to be here. I know a lot of people in the audience, so every time I give a lecture here, it's a lot of fun. And Woods Hole is just such a special place. So I'm going to tell you about stem cells, and at the end, I will play the double high C for you. So there is a... So I founded a few biotech companies, and I won't discuss what they're doing. So I've spent most of my entire life studying blood stem cells, also known as hematopoietic stem cells. I'm a hematologist, and I take care of patients at Boston Children's Hospital. So if you think about inside your bones is the marrow, and that's where the place where blood cells are formed. And there's a constant influx and efflux of hematopoietic stem cells. And these stem cells have the ability to renew themselves, or they can differentiate into the blood cells. And we know this process works very well because you can do a bone marrow transplant on a patient and rescue them from a near lethal dose of chemotherapy, and one transplant is enough to rescue them for their entire life. But still 25% of patients die from the transplant. Blood stem cells are the only stem cell which has a quantitative assay, and that is the transplant. Blood stem cells are the only stem cell besides skin that are used therapeutically. There are thousands of bone marrow transplants done every year, and hundreds of gene therapy and gene editing trials use blood stem cells. And when a mutation happens in a blood stem cell, this can ultimately lead to leukemia. So, you know, Tatiana mentioned that we do all of our work in zebrafish, and I thought I'd spend about a third of my talk telling you how an MD actually got into doing zebrafish work. And so I'm always asked, how did you get into zebrafish? When my lab started, we actually were working on frogs. And I went to this meeting in 1992, which is called the Hemoglobin Switching Meeting, and I presented my work on frogs. And there was a scientist there, Frank Grossfeld, who's a famous mouse geneticist, and he took me aside and he said, you know, I really loved your talk. He said, but have you heard about the zebrafish? And I said, wow. He says, zebrafish genetics are really great, and you should think about switching. And, you know, every so often, when you meet somebody at a meeting, you know, you can have a quick conversation, but this was an hour-long conversation and really had an impact. So I came back from the meeting, and the next day, a scientist, Bill Dietrich, who works on the Antarctic ice fish. He spends 3 months a year in Antarctica, and the fish swam down there and then lost its blood cells. The reason is that it strokes out from being too viscous, at 0 degrees temperature. So he wanted to come and do a sabbatical in my lab to study how the ice fish deals without blood. And I said, I didn't really care about that, but what about zebrafish? And he said, okay, sign me up for a sabbatical. And then literally the next day, Wally Gilbert's lab called, and Wally had won the Nobel Prize for sequencing, and he said, you know, we have a zebrafish mutant that we made that has no blood, and we're going to throw it out, but you know, we thought you might want it. So I said, sure, we'll take it. And so within 1 week, somebody upstairs was telling me you better do zebrafish. So this is a zebrafish. Zebrafish is about 1.5 inches in size. You can buy it at a pet store for $1. And at Children's Hospital, we have 300,000 of these fish. And that's almost at a level that you can do population genetics. And the zebrafish is really fantastic. The embryos are completely transparent. We can see them right at a light microscope, all the different organs developing. You have circulating blood within 23 hours. And we've made a transgenic fish where we've labeled each type of blood cells in a different fluorescent color. Then each mother has between 100 to 200 babies every week. We say childcare is a little bit expensive for the fish here. You can do genetic screens, you can do genetic knockdowns, you can add chemicals to the water and cause interesting things to happen to the fish, and the blood program is amazingly conserved. So this is a 4-cell embryo, and what you're gonna see is it turns into 8 cells, 16 cells, 32 cells, and it's sitting on top of a yolk. The yolk is the nutrient supply for the fish. Now over on the right side, you'll see the developing brain. So this brain starts over here, and then it curves back up, and then it's going to start at the top here to make an eye. Okay, and you'll see the eye develop right here. There you go. The muscles are forming, and the tail lifts off. And all of that took 19 hours. So it's incredibly fast. And when we look at a 19-hour-old zebrafish embryo, it compares very, very similarly to a 28-day-old human embryo. And so when I started doing this, the hope would be that the zebrafish would tell us some things about human biology, and that turned out to be true. So, here I am, and it's in the early 90s, and I, really excited about zebrafish. It's got great genetics, excellent developmental biology, it's externally fertilized, we can add things to the water. But Don Wiley, who's a famous X-ray crystallographer, told me, it's easy to get talked out of a good experiment. And I thought everybody was going to be extremely supportive of me doing this. But actually, my colleagues had a lot of questions for me. They weren't so supportive. If you're going to try to make mutant fish, how many mutants are you going to find? Will you ever be able to clone the genes for these mutants? Will you ever find anything new? Or will you ever find a gene that regulates stem cells or even blood cell development? And so these were really penetrating questions, very difficult for me to figure out. But we went ahead and made zebrafish mutants. We could take a chemical called ethyl nitrosourea. This causes point mutations of the sperm and spermatogonia. We would then mate this to a female fish and generate a family of fish that are carrying a lot of mutations. Then we'd mate this fish and then generate the 3rd generation and look for mutations that occur in an autosomal recessive pattern, where 25% of the offspring have a genetic disease. And in our case, they don't have any blood. So here's a normal fish, and this is the heart. And if we look here, you can see the heart pumping a pink fluid through it, and that's blood, okay? So then we saw this thing, which is a mutant, and it's got blood cells, but not the pink color. So this fish has the equivalent of sickle cell anemia or thalassemia. So it's a genetic mutant, a genetic disease that's affecting the blood system. And then we had this mutant, which had no blood. It was pumping, but it has no stem cells, so it can't make any blood. And that was very exciting for us. It was pumping plasma all the way through the circulation, but no blood. So, at this time, the zebrafish field, we could kind of order these along a continuum from the beginning of embryogenesis all the way to making red blood cells, white blood cells, and lymphocytes. And so we had early mutants. We had mutants that affected blood stem cells and these things called progenitors. And then we got red blood cell mutants. So this was pretty exciting. And at that time, you got to name your mutants. And so we decided, because there's red and white blood cells and red and white wines, we would name our mutants after wines. So we had Zinfandel, Chianti, Chardonnay, Chablis, Riesling, Sauterne. So this was going really well. And what I would do is, whoever cloned the gene, which took a couple years at that time, I would buy them a very expensive bottle of wine that represented the name of their mutant. So when Alison Brownlee first cloned Sauterne, I bought a $100 bottle of Sauterne for Alison. It was super exciting. So everything was working well until I came up with Barry Paw cloned this gene, Retsina. So I never heard of this wine before. So I went to the liquor store and I said, I want your best bottle of Retsina. And the guy says, do you want the gallon jug or the half gallon? So I didn't know, that Retsina was a cheap Greek wine, but, could be tasty. Then we found T-cell mutants, mutant fish that had no immune system, had no T-cells, and so we named those after teas. So we had chamomile, Darjeeling, Earl Grey, jasmine, and Assam. Okay. So I put this quote in from Yanni Nusslein-Volhard. So we collaborated with Yanni's lab. Yanni won the Nobel Prize for understanding how embryogenesis occurs in fruit flies. And she then switched her lab to zebrafish. And my lab wanted me to pick up some mutants in her lab. And she had invited me around 2000. And so I went over, and she arranged to have all these fish picked up in the fish house. So I show up at the fish house. I don't know what I'm really doing because I haven't worked directly on these fish before, but they have a duffel bag that's like this big, and it's filled with 50 flasks of adult fish. And I'm supposed to bring these back on the plane with me, okay? So all night long, I can't sleep. And I go into the Stuttgart airport, and the first thing that happens is you have to go through the X-ray detector. And I don't know, like, can the fish be X-rayed? Like, I don't know, you know? And so I put them through the X-ray machine, and then the guard on the other side says, I must inspect your baggage. The baggage was huge, right? So I said, okay, and he opens it up, and he holds up a flask, and he goes, oh, fish. And he says, go ahead. So, so I go through the line, and so now it takes 2 compartments for this huge duffel bag, right? And an hour into the plane ride, I said, I better check on the fish. So I open up the container, and basically it's completely wet. Completely wet. So I look inside, and 3 tanks have cracked. So I don't know what to do, so I push my button, and I say to the stewardess, I said, I'd like a napkin, please. And so she comes with 1 napkin, the little thing. I said, no, I'm going to need a few more. And so every hour I would pat down the thing. And so then they come with the card at the end, which says, are you bringing meat or fish into the country? I was trying to figure this out, and so I decided I was so fried from this experience, I just checked the box. So now I go all the way through, and I go through customs, and the guy says, it says here you're bringing meat into the country. And I said, no, fish. And he says, okay, go ahead. So anyway, the fish made it back, and even the fish that with the tanks that had broken made it back, which was nice. Okay, so we have all these mutant fish. It's super great. And we have to ask how to clone the mutant genes. At this time, the zebrafish genome was 2/3 the size of humans. There were 25 chromosomes with 1 marker per chromosome, and there were no libraries of zebrafish DNA. And I knew I was in trouble. It's going to be super hard to do this. And so I had a conversation with a famous hematologist, George Stamianopoulos, and he says, Zon, you're never going to clone these genes. And they're like, yeah, you're right. He says, the only way you clone these genes is if Harold Varmus sets up a zebrafish genome project. And I'm like, wow, that's genius, because I know Harold. Harold was the NIH Institute director. And so I said, I'll just call him. So I called him. And he said, have a meeting, write a white paper. But he says, I suggest you speak very briefly about the benefits of zebrafish, since some of the directors are unfamiliar with them. Pass out copies of the report for leisure reading. Describe the obstacles to progress. Outline the major recommendations for building the infrastructure that's lacking. Plan for talking for 20 minutes. Don't expect any instant decisions, but I think you'll have an impact. And so I went down to give a talk for the zebrafish community. I had all the best slides. I was super nervous. And I walk in the room, and Tony Fauci is there. He goes, Len, I used to breed zebrafish when I was a kid. And they're like, all right. So all good. And we have the Trans-NIH Zebrafish Genome Initiative. And then we were able to basically clone all the genes. And as Tatiana said, about 5 of these genes were novel at the time. And then we found humans who were mutated in the same genes as the fish. And so we had discovered 5 human diseases as a result of this big experiment. So, being a fish biologist can come in handy sometimes. And so, as Tatiana said, I started this International Society for Stem Cell Research. And in the 10th anniversary, which was in Yokohama, my friend Shinya Yamanaka, pictured here, gave me a call about 2 weeks before the meeting and said, I have a treat for you. You're going to give a talk at this meeting, and the Emperor and Empress of Japan are going to come to your talk. And I said, oh, that's really great, no problem. And he said, the only thing is I need to teach you how to bow. So before this, he gets me in a closet. And I mean, it's a closet basically the size of this. And he says, all right, start to bow. And he goes, no, 30 degrees. If you go 45 degrees, it's an insult. If you don't go to 30 degrees, it's an insult. So you got to hit 30 degrees. So we practice. And then as he's walking out the door, he says, one more thing. Do not touch the emperor. So I go, okay, that's fine. So now I have to bow first, give my talk, bow again, and do everything well. And then the emperor and I end up in a room together. And the emperor sticks out his hand. And I didn't know what to do because, should you touch him or not? But I shook his hand. So so then my graduate student told me the emperor of Japan is a fish biologist. So I looked it up online, and sure enough, he has a science paper. Now, when David Skadden and I write a paper, it says, Harvard University or Mass General or Children's Hospital. His says the Imperial Palace. So anyway, I hope I've convinced you that zebrafish are cool. And now I'm going to go a little bit into the science part. And this is a movie of how stem cells are born in the zebrafish. So this is a 36-hour embryo. And we're going to dive into the aorta of this fish, which is the largest blood vessel of the body. And that's where your blood stem cells are made. So what happens is a blood vessel cell, an endothelial cell, changes its fate. It rounds up to become a blood stem cell. That stem cell could be transplanted if necessary, but in the animal, it goes into circulation. And it's going to go around and around circulation looking for a place to land. And it will land in this tail region in the zebrafish. In humans, in the fetus, it would land in the liver, in the fetal liver, and make that a blood-forming place. So this stem cell stops on a dime and transits into what we call the niche, the environment of the stem cells, where it starts to double. And then some of these cells go off into circulation and are going to colonize the marrow. Now, the marrow in a zebrafish is in its kidney. In humans, it's inside your bones. But this makes the kidney a hematopoietic organ in the zebrafish. Some of the cells bypass the kidney and go to the thymus, which is the beginning of the immune system. And all of this allows this animal to have blood really throughout its entire lifetime. So several years ago, we made a fish that had fluorescent stem cells, green stem cells. So the stem cells are born in the aorta, they go round and around circulation, and they land in the tail region. When they land, you can see that the stem cell is here, transits outside the blood vessels, but then weirdly we saw that the blood vessels wrap themselves back around the stem cell. We called this endothelial cuddling. Then there's a division of the stem cell, a turn, one stem cell leaves and the other one stays. And this is a very stereotypic behavior. And we think this type of thing happens when we do a transplant in a patient. So we took that animal and we did something called correlative electron microscopy. And we were able to solve the highest resolution structure of a niche of the environment of a stem cell. And we saw some interesting things. The stem cell was physically attached to a specialized fibroblast called a stromal cell. And it was surrounded by 5 blood vessel cells or endothelial cells. And so that's the structure. And so we've been trying to work on this for many, many years. So because we saw this niche was so interesting, we decided to take a 3-dimensional view of this niche. And we did something called RNA tomography or tomoseq. This is a zebrafish embryo. And this particular region, we started to section across. And every section was put into a tube, and the gene expression in the section was analyzed. So you could see here, for instance, the spinal cord was up near the top of the animal, but there were a number of genes that were only present near the stem cells in the niche. And we started to look at some of these genes. Many of these genes were in the blood vessels that were surrounding the stem cells. These are specialized veins. And they're even more specialized veins. They're called, sinusoidal vessels, venous sinusoids. So we can do gene expression using a technique called whole embryo in situ hybridization. It's just where the RNAs are localized. So here is a gene that's expressed in all blood vessels, and you can see all the blood vessels of this particular fish. These 4 genes were found to be those ones that are only in the cells next to the stem cells, and you can see them here, there, and they're very specific. That's the only place that they're expressed. So we started to understand this a little bit better when we took the genome regions upstream of these regions and coupled them to green fluorescent protein and made transgenic fish with these regulatory elements. And here you have a little piece of DNA, and this piece of DNA is able to drive expression just in the niche endothelial cells, just where the stem cells are around. So with that, for the first time, we could actually sort those specialized veins using a flow sorter and then study their characteristics. And one of the things that we did is we looked at their DNA and how it was packaged, the chromatin, and we found regions of the genome which were only open in the venous sinusoids, but not in any other blood vessels. And this is one of our favorite genes, MRC1A. It's a mannose receptor. And when we took this little particular region, which was 125 base pairs, and made a transgenic fish, the only thing that lights up are the veins next to the stem cells. So with this type of approach, you could compare across a lot of different regions that have the same ability to just be open in venous sinusoids. And we found motifs of DNA using some algorithms, and those motifs could bind certain transcription factors. And we thought that these particular DNA binding proteins could be used to reprogram an ectopic niche. So in other words, making a brand new place where blood cells would form. So this worked incredibly well. These are just some regions here that we've picked. This region here in this stabilin 2 gene and another one in a legumin gene. And whenever we made transgenics, they light up this region where the veins are. So we were able to find a set of genes that could turn on this program and bind to those specific DNAs. And so we have here members of the ETS family, the SOX family, and nuclear hormone receptor family. And we wondered if any of these could be used to reprogram an ectopic niche. So similar to what Shinya Yamanaka had done for stem cells, we started to overexpress these transcription factors. And when we did this, we saw that the fish developed extra blood vessels that shouldn't normally be there, but it starts making new blood vessels. And you can even see it with our transgenic fish where they're fluorescent blood vessels, extra blood vessels there. And we were able to find 3 transcription factors that did this really, really, really well. So then Elliot Hagedorn, who taught in the Woods Hole course here, came to me one day and he said, "This is the best day I've ever had as a postdoc, ever." And I said, "Well, what'd you do?" And he says, "Well, I injected these 3 genes into a fish that has red stem cells. And I made all these new blood vessels. And the stem cells that should be where they normally are traveled to the new place. So it started setting up new blood development." So I got excited, and this is a high-resolution picture of all the stem cells next to these vessels. So the stem cells were finding these new niches. So I said, "Well, did they cuddle?" And he says, "Yeah, they cuddle the stem cells." So then I said, "Well, did the stem cells divide in these new locations?" He goes, "Yes." So this is a fish, and you can look at this box. It's represented by this movie. And this is the normal place where the stem cells are. And you can see all the red stem cells next to the green endothelial cells. But up here is a set of vessels that shouldn't be there. And you can tell they've already attracted the red stem cells there. And if you just focus on this region, when I play the movie, you'll see that those stem cells are dividing in that location. So we've fully reconstructed a niche, in an ectopic location, and this was very, very exciting. So one question is, this was a fetal niche or a larval niche, and we wanted to know, what about the adult niche? And what I can tell you is the same genes are expressed in the adult niche. And then we collaborated with a laboratory in Cornell, Shaheen Rafi's lab, and we studied mouse blood vessels. And we took them out of these different organs during fetal life or adult life. In the fetus, the liver was very high in the number of genes that we were studying. And remember, the fetal liver is the site of blood development in a mouse. But then in an adult, it switches to the bone marrow, and that's where these genes are. So now we have a method to make these new blood vessels, and you could say, what's the use of this? Well, there are patients who have a disease called myelofibrosis. It's actually very common, particularly as people age. And what happens is the marrow fibroses over. So the fibroblasts take over, and the stem cells have to leave, and they attempt to find a new place to make blood. Some places in the liver and spleen is where they go, but it's a very inefficient process. So if we could actually take stem cells from humans, iPS cells, and put in our transcription factors, we'd be able to reprogram them to these endothelial cells. We might be able to inject them into the liver and give a better place for all these stem cells to start dividing, or we actually might put them in the arm, and you might start making blood in the arm, and that would be would rescue the patient. And so these are things that we've actually been able to do, at least in terms of making the iPS cells turn into these endothelial cells. So it's things that we're hoping to do in the future. So now I'd like to talk to you about bone marrow transplantation. And you know, I'm always asked, like, what happens in a bone marrow transplant? So unfortunately, we're all opaque, so I can't tell you. But what happens is it's a little bit anticlimactic to do a bone marrow transplant in a patient. I show up at the bedside. There's a bag. It looks like a bag of blood, but it's a bag of marrow. They're both red. And then I just start an IV, and I put the stem cells into the veins. And the stem cells know where to go. They go to the bone marrow. And this is a process called homing. So homing happens usually within 16 hours. It's very, very fast. So the stem cells know where to go. But then there's this process called engraftment. And I'm always asked, you know, what is engraftment? So think about when you were in college or university, right? And you took your final exams, okay? And then you went home. So what'd you do for the first 3 days when you're home? Sleep, right? Okay. And then in day 4, you wake up in your own bed and you have that feeling, right? So that's marrow engraftment, okay? And then a single blood stem cell, one blood stem cell can make all 6 pints of blood that you have in your body. And remember, it's only needed to be done once. So it's an amazing process. So, you know, around this time, in the early days, I wanted to put my whole lab on doing a marrow transplant in a zebrafish. And I figured, I don't know much about this, but I'm going to ask somebody who does. So I wrote a letter, this was before email, I wrote a letter to E. Donald Thomas, who won the Nobel Prize for bone marrow transplant. And he wrote me back, and I was super excited. This is the letter I got. And he wrote me that he's already done the experiment in 1960. So now remember, I wanted to put my whole lab. He said he thought it was a good project for 2 high school seniors. Wanted a science project. We got some zebrafish, learned how to get the kidney marrow in a sterile condition, did a few injections, presented it at a science show, but it was quickly too complicated for him, and he went back to the canine marrow transplants that won the Nobel Prize for him, and the fish project was dropped. You know, it's really hard to do a novel experiment, you know? So the modern day version of this was we had the fish that had the green marrow, and we could transplant it into an irradiated adult. And when we do this, all the blood was green. So this was super exciting. And you know, what I'm showing you here is the fin of the zebrafish. The fin is so thin that you can image through it. But I wanted to see the stem cells go into the kidney. And unfortunately, the stripes of the zebrafish were getting in the way. So one of my postdocs invented this fish called Casper, the transparent adult fish. And this was a couple pigment genes put together. And, you know, you can see the eggs here in this fish. You can see the liver. You can see the aorta. You can see the bones. You can read the newspaper through the fish. So this was pretty exciting. And around this time, the Wall Street Journal did an article about our fish. And so, you know, it was pretty exciting. But I got this email from a friend. It says, "You are famous." And I said, like, what happened, you know? So what happened was...
Children's Hospital in Boston have genetically engineered transparent fish, see-through fish. The fish are totally transparent. No, not amazing? And you know Red Lobster's going to jump all over this for their seafood platter. We didn't forget your fish. It's just invisible. They're right there. Just look
So there you go. So
You'll
zebrafish research can make you famous. So, so I wanted to tell you about some of our modern day studies of stem cell clones. And this work really made us think about how transplant is a good system, but it's also a little bit inaccurate. So when you transplant a stem cell into an irradiated host, the host is inflamed, and the transplanted cells are stressed, and only some clones of stem cells will actually go on to contribute to the transplant. So we and a number of others have developed some systems for cellular barcoding zebrafish. What this means is to use DNA as a barcoding system to follow the cells. And there's two versions right now, but many versions coming up. One is to make to color each stem cell a different color using DNA. The other is to use CRISPR, a technique which will genetically engineer certain barcodes into the fish and follow these lineages. And in this particular case, you just bleed the animal monthly, follow the barcodes, and you can tell how the clones of stem cells are doing. There's no transplant, no stress. The stem cells are in their native environment, and it's a real accurate assessment of clonality. So one of the reasons that I wanted to study, use this technology was to study this disorder called Clonal Hematopoiesis of Indeterminate Potential, or CHIP. So this is a strange thing. About 10 years ago, if you would have asked me, how do we age? I would say we age through epigenetics. Okay, so epigenetics means you don't really understand the process. So, but it turns out that that people sequenced the blood of old people, and they found mutations. And they found recurrent mutations in about 20 different genes. And when they saw this, they found that a single stem cell in these people were giving rise to 20 to 30% of the blood. And this was predisposing them to myelodysplasia and ultimately leukemia. And this is unbelievably common. It's estimated that 12% of all 70-year-olds have mutations in their blood cells. And this is very, very common. 30% of 80-year-olds and 50% of 90-year-olds have mutations that are driving the blood cells. And this is also associated with cardiovascular events like clotting and heart attacks. And so it's thought that maybe some people are getting heart attacks and maybe due to this mutations that are happening in their blood system. So we wanted to study this process, and we used the Zebrabow system. So this is a zebrafish that has 15 to 20 copies of this plasmid distributed throughout its genome. This is red, blue, and green, separated by a recombination site. When you mate this fish to a blood-specific line that has the recombinase, you start turning all the different colors of the rainbow. You remember the old RGB TV sets that would dial up all of the different colors, so this is the same type of thing. So we could add tamoxifen, turn all of the embryonic blood different colors. Then what happens is the embryonic blood is removed eventually, and the stem cells that are colored will make adult blood. And you can look at the number of colors in the blood and back calculate how many stem cells were born in the embryo. So this worked really well. This is an animal that has multicolored blood, and you can see it traveling around. And if you look over here at this particular cell, that particular cell is dividing like a stem cell. So that blue-green cell, when it grows up to adulthood, will make blue-green blood. So now we grow up the animal for 4 months till it's an adult, and we bleed it and do a blood smear. And that's the blood smear, okay? Now, I'm a hematologist, and I can tell you that's the prettiest blood smear I've ever seen. So we were able to calculate that at a certain time in development, there are 21 clones of stem cells. So we wanted to understand more about clonal hematopoiesis, this disorder, and what's known about it is a stem cell gets a mutation. But the genes that are mutated really don't lead to much problems. They make neutrophils, the white blood cells, and macrophages very well. But interestingly, this causes the macrophages and neutrophils to pump out inflammatory growth factors. And this makes the entire marrow inflamed. Then by an unknown process, you get expansion of the stem cells, and then new mutations lead to leukemia. So Serena Vagian in the lab, a postdoc in the lab, invented a technique called Twister. And yes, this was a play on the Twister game. She injected guide RNAs and Cas9, so this is CRISPR, for the 20 most common genes that cause the clonal hematopoiesis. And she did this in a way where half the stem cells were mutated and half the stem cells were normal. And they could compete against each other in the same animal without doing a transplant. Then we did our color trick, and you could just bleed the animals based on the colors. And what's exciting about this is we can sort with a cell sorter the dominant clone and compare it to the non-dominant clone in the same animal. And so we'd understand more about the mechanism of the disease. This worked very well. The control animal has many different colors. The mutant animal has a dominant clone. 60% of the blood is coming from one stem cell. I'm going to skip this. So when we did this, we could see that the mutant pool was leading to cluster size differences, clonal dominance. And there's another way, an economics term, a Gini coefficient, to actually show the same thing. So in our system, this particular gene, ASXL1, led to a lot of dominant clusters. And this is what it does in humans. And about 10 to 11% of all humans that have this disorder have this mutation. So then we studied the RNA expression of the different populations, and it's summarized over here. What you can see is that those mutant neutrophils and macrophages were making those inflammatory growth factors. But what's interesting in the stem cells, they actually, we found, they were making anti-inflammatory factors. So what's happening when you have a mutation like this, it does two things that are very strange. For the mature cells, it makes them inflamed and pumping out inflammation, but it makes the stem cells resistant to the inflammation, which is why over time they would actually accumulate. So then we used our Twister system to actually do genetics. We used CRISPR here to knock out this gene, ASXL1, and we saw clonal dominance. But when we knock out one of those factors that was an anti-inflammatory factor, we thought that this would block the expansion of a clone. And that's what we saw. So if you are a homozygote mutant for this particular gene, you don't have as large a cluster size as you do without. And interestingly, when we did this particular perturbation, there was no effect on normal stem cells. It was only on the mutated stem cells. And so this would suggest that if we had a drug for this, it would be a therapy for these people. So just to summarize this, what we found was stem cells that are mutated can go on to make these normal neutrophils and macrophages, but they will express inflammatory growth factors. This upregulates a response in the stem cells that's anti-inflammatory, and it leads to this enhanced growth. But if you knock out this particular transcription factor, you stop to suppress. And we think that's an interesting mechanism. And now we have some antagonists, actually, that we've tested in the fish, and they do suppress the clonal dominance. So we're hoping to take that to the clinic. Okay, so I want to finish with a short story, hopefully, on macrophages. So when we were looking at the macrophages, now in purple, that were in our stem cell niche, we were surprised to see that they were really intimate with the stem cells, and they were removing material from the stem cells. So Sam Watrous was able to study this more in depth, and what he saw was about 20% of all the stem cells that had just been born were completely eaten by the macrophages. Whereas most of the time, the stem cell interacted with a macrophage, a little bit of the material was removed from the stem cell. And then within 45 minutes, the stem cell would go on and divide. It was almost as if the macrophage interaction was a licensing event for the stem cell to divide. So this was very specific for the stem cell macrophage interactions. There wasn't a lot of blood vessels or red blood cells interacting with the macrophages. And when we look, about 75% of all stem cells in the body were interacted with a macrophage. So in the fish, we have some really cool tools to get rid of the macrophages. One is to give this drug clodronate that kills all the macrophages. The other is to use a genetic approach to knock down this gene which depletes the macrophages. So because we saw a cell division thing going on in the embryo, we decided to evaluate the number of stem cell clones. And so what we did is we injected this genetic perturbation, reduced the macrophages, or we could give this drug that kills the macrophages. But then we did our color trick to assess clonality in adulthood. And what we were surprised to see, if you get rid of macrophages, you have about half the number of stem cell clones in adulthood. So this means that there's some quality assurance mechanism going on, and the macrophages are trying to figure out what I don't like about the stem cells and how I'm going to take some of them out, and some of them I'm going to cause to proliferate. So one of the things that's cool about this is this green macrophages that had eaten red stem cells could actually be purified because they still have a little bit of red in them. And that allowed us to do some really interesting comparisons. So first of all, we were able to find a bunch of different macrophage populations, and it's this particular macrophage population that's the one that's eating the stem cells. And this is very much enriched for lipid metabolism and fatty acid metabolism. So these are the ones that are doing it. And we even found markers like HMOX1 that's a marker of these particular macrophages that are eating on the stem cells. So the thing that we were trying to understand was the mechanism. And so we worked with a laboratory at Harvard that could do something called few cell proteomics. They can do proteomics on 120 cells. And so we submitted the macrophages that had the red against the macrophages that didn't have the red. And what we found was there were 3 paralogs of a particular protein called calreticulin that were differentially regulated. Calreticulin is a ER protein, and it has this sequence that makes it retain in the ER. But in times of stress, it can go to the cell surface and decorate receptors on the surface. And Irv Weissman's lab has been able to find that calreticulin is an eat me signal for cancer. And it works through a receptor LRP1, and so this is how cancer cells get eaten. So our particular macrophages were very high in LRP1, so we thought that this could be one of the mechanisms that's going on. So we actually made a calreticulin construct that could overexpress and get out of the cells, and there were more macrophage stem cell interactions. And this is quantified here. With any of the 3 paralogs that we overexpressed, you would get more macrophage interactions. So then what we did is we wanted to see if you knock down calreticulins, what happens to clonality? So first of all, we measured macrophage stem cell interactions. If you knock down these 2 paralogs, you get less interactions. And we also looked at clonality with our color system, and you get less clones of stem cells. So that tells you that the calreticulins, particularly 3A and 3B, are required for this macrophage stem cell interaction. But it doesn't tell you which, is it on the stem cell side or on the macrophage side? So to study this, we made fish that are parabiotic fish. So what we did is we joined the circulations of 2 fish. And on this side, we had a fish that has red stem cells with calreticulin knocked down. And on this side, we had a fish that has green macrophages. And when we look at this situation, there are less macrophage stem cell interactions in this particular conformation. But if we switch the colors and look and knock the calreticulin down on the macrophage side, there's really no effect. So it's very specific. The calreticulin must be on the stem cell side. Then we measured cell division. We have these zebrafish that fluoresce a certain color when they're in a certain phase of the cell cycle, G2M, and the macrophages love to interact with these cells that are in G2M, and that's just quantified here. And again, it's within 45 minutes after the interaction that the stem cells will divide. And we also labeled the stem cells but with a technique called EDU to look at the proliferation of those cells. And when we knocked down these factors, you saw a reduced number of divisions. So this sets up a model where basically in the normal animal, the cells that are born in the aorta end up being represented in the adult, the clones. But here, when you have fewer cell divisions, if you get rid of the macrophages, there's kind of an asymmetric divisions here. And this will lead to a situation where certain colors are actually favored, and that's why there's less clones of stem cells that are actually participating in adulthood. So this is a summary, is just that the stem cell can come poke through, and then one of the things that happens is it's greeted by a macrophage. And in about 20% of the cases, the macrophage is going to come along and just completely eat the stem cell. So there must be something that it doesn't like about that stem cell. Whereas most of the time, the macrophage will come along and will interact with the stem cell and just take a little bite, okay? And when it takes a little bite, somehow it causes the proliferation of those stem cells, and those stem cells will execute kind of a characteristic behavior where they'll attach to the stromal cells and then divide outward. So all of this makes you wonder about what is it that they're detecting. And so it turns out that we think there's a lot of stress when a stem cell is born. And it might develop reactive oxygen species or DNA damage or ER stress. And if there's a lot of calreticulin on the surface of a stem cell, it's going to be completely eaten. If there's an intermediate level of calreticulin on the stem cell, it's allowing the interaction. And we think that the macrophage itself is able to actually bring in some growth factors and stimulate the production of the stem cells. And I should say calreticulin itself is known to be mutated in cancer and causes a myeloproliferative disease. So with that, I just want to thank the people who've done the work. I think I mentioned everybody's names, so I won't go through all these, but we're very thankful for a wonderful lab. And then I want to show you on the last, I always believe as a trumpet player, ending on a high note. So during COVID, it took me 1.5 years, but I was finally able to play a double high C on the trumpet. So I will prove it right here. Thank you very much. Appreciate it. Happy to take any questions.
Thank you very much. That was fascinating. So do you think that the 25% failure rate is due to the macrophages eating the stem cells 100% or due to the stem cells not forming?
Yeah, so what we really think is, so the stem cells, there's a code to be eaten. So there's an eat me signal, and then there's a don't eat me signal. And we've actually done a chemical screen, and we have chemicals that can force the eating behavior or the division behavior. And so it's kind of having 2 or 3 signaling pathways that are triggered to allow one event over the other. But this may be extremely helpful to try to remove bad clones. So we're trying to teach the macrophages to remove the bad clones, and that would be a therapy for some patients. Thank you for the question.
Hi, thank you. I was wondering, the little bit that gets eaten off, is there a way to isolate that to see if there's what that signal might be or what it's removing in order to
yeah, so we've been looking at this. So one of the things that's fascinating, when we did our proteomics studies, there was the calreticulin there, but there was also a set of proteins that are involved in something called exosomes. And so exosomes are actually transferred from one cell to the other, and they can contain RNA or protein or even mitochondria and other things. So now we're looking at what is the material that's actually going to be transferred. We don't have any definitive proof of any single model, but we have some intriguing data that we're trying to follow up. So that'll probably be the next story. Thank you.
Is there any difference in the stem cells that lack the macrophage interaction?
Yeah, so almost all macrophages will interact with a stem cell, and it's very surprising that it's that high, to be fair. So we see 75% by direct imaging, but that's very imperfect because a macrophage can move and quickly come back into the circulation and come back again and do it. Yeah, so we have stem cells. So those animals, no. So basically, when we've looked at the animals that have their stem cells knocked out, they have a normal differentiation pattern, but they have a higher level of reactive oxygen species. So we think one of the signals that's being detected is this, oxygen radicals that build up. And so that may be one of the reasons to remove the stem cells, just too much of these, of ROS. It's very interesting. We're following up some of these. When we give antioxidants, we can kind of prevent some of the macrophage stem cell interactions. So it's interesting.
On your list of genes implicated in stem cells becoming dominant clones, I noticed a lot of DNA methyltransferases. Are there distinct methylation patterns between the ones that become clonal dominant and the ones that don't?
So this has been one of the conundrums of the field. There's no consistent patterns for methylation changes. It is true for the DNMT3A mutated, they have a methylation problem, but it's not consistent between all the other types of clonal hematopoiesis. So although methylation probably plays a role or may have a role during stress activation of genes, at steady state, there isn't that much of a difference. And it's one of the conundrums that the people studying human blood cells have had. They really don't know why these patients have these clonal hematopoiesis changes.
Thank you for your talk. Does this mechanism also apply for adaptive immune system, particularly B cells?
Yeah, so right now it seems mostly an innate immune system issue to recognize the stem cells, but there's been some recent work demonstrating that certain T cell classes can be very important for clonal hematopoiesis. So it's a brand new area. We were going to look at that, but then we found the macrophages were super important, so we've been studying those rather than the T cells. But it's a good point, and it's possible that the immune system is suppressing clones of stem cells and activating certain clones, and the adaptive immune system could be involved in that process.
I loved your cuddling story at the beginning. Can you tell us anything about what the attractants are that those endothelial cells that are attracting the stem cells back to the ectopic sites?
Yeah, so that's a topic we're hot on the trail of. We don't know the mechanism of the endothelial cuddling. We have drugs that can stimulate endothelial cuddling, and they maybe are a little toxic, so it's not clear how direct they are. But recently we've been looking at lipids and things that have been involved in endothelial biology, and there might be something there. So that's what we're chasing down right now. But we don't really know what the attractants are. SDF1 is required for the stem cells to actually bind, but that's not the thing that's causing the cuddling. So there's something else, and we need to do genetics to understand it. Great question.
Back deck of Swope, and please thank Dr. Zahn.